have started about the immunology module and the defense mechanism, external defense, internal defense, immune response, the primary immune response, secondary immune response means the uh, innate immune response, acquired immune response. What about antigen, antibody? We have got already a overview. Now this one also has been covered. What are the cells participating in the immune system? Achha, I couldn't find today the class recordings.
Recording has started.
Today it has been started. The previous class recordings, when I just revisited your files, class materials recordings, no recording was there. Why I didn't know. We recorded everything. So immunological memory, have we completed immunological memory? Let me start from the immunological memory. The ability of immune system to respond more rapidly and effectively to specific pathogen means if one pathogen is interacting for the first time, the cell participating in the immune response, mostly the bone marrow
Wait,
generated.
ma'am.
Huh, huh.
Ma'am, the previous class we did it till vaccination protects us by infection. That part, like slide number I think 16 or 17. We didn't do
That's
colonal
why
section.
I was searching the previous record, but no recording, class recording I could found. Why they are not there? I can't understand.
Yes,
Oh,
ma'am. From
then,
here we start colonal section. That this
so
thing we have done.
this one, the vaccine protects us from infection by inducing the adaptive immune response. It has been cleared, no? So from clonal selection of the lymphocytes. So lymphocyte, monocyte, I will be explaining later. First, go to the antibody. I am coming in the slide number 16. Second, first go to this one, immunological memory. The memory, when the first time the cell systems interact with the pathogen, one population of the cell may be the B lymphocyte or the T lymphocyte. Both are differentiating into two populations. One, two, the effector population, which will be effectively giving rise to the immune response, and another population is the memory population, which will be carrying the memory of the same antigen for the futuristic use. Now, the adaptive immune response, it comprises with humoral immune response as well as the cell mediated immune response. Humoral immune response, it involves mainly the B cell driven antibody mediated immune response, and the cell mediated immune response, it mainly comprises with the cytotoxic T cell and helper T cell mediated immune response. So, if you just consider the immune recognition molecules for adaptive immune response, means the which are the main causative agents which are responsible for recognizing the antigen, because antigen is the non-self product. When they are interacting with the in vivo system or the cell, then the cell can't recognize them very easily. At first, cell has to interact with the antigen. Now, the antigen wholly is not interacting with the host cell. The epitope or the specific site of the antigen is interacting with the receptor of the anti host cell. Now, host cell express some specific receptors called the B cell receptor or BCR. Antibodies are also acting as receptors, and T cell receptor over the T cell and the major histocompatibility complex, another polymorphogenic gene-derived protein, which are responsible for recognition of the epitopes of the antigen. Now, the antigen-antibody interaction will give the B cell triggering, and this B cell, you see, this is the B cell. If there is no interaction with antigen, then the B cell won't survive longer and multiply in number. But if there are need, interaction of antigen is the need, and the result is the production of antibody. So the production of the antibody by the B cell is facilitated, and the plasma B cell, it produces antibody, and you just see it is called the effector B cell. Now, one population of the B cell differentiate into the plasma B cell and take part in the humoral immune response. Another population of the B cell will be kept as the memory B cell for further use, for further recognition of the same antigen. Now, the B lymphocyte, they originate in stem cell of the bone marrow. From the stem cell in the bone marrow, they mature also in the bone marrow. And whenever they mature and they have to migrate to the secondary lymphoid tissues, means in the distant region of the body, there are lymphoid tissues. Let's see this picture. There are different lymphoid tissues: tonsil, adenoid, the thymus, then the spleen. There are small lymph node in the groin region, in the shoulder regions. So these are the peripheral lymphoid organs, and the blood which is given rise in the bone marrow, the pluripotent cells, they can differentiate into B cell, and the B cell also mature here. But the T cell, they generate, they develop in the bone marrow, and they migrate to the thymus here to mature. That is why their name is T cell. So both the populations of the cell, they come together in the distant lymph nodes for the exchange of the antigenic information, because the B cell, they help the T cell to get charged. The T helper cell, they help the B cell to get charged. So all the cells, they take part differentially and act in a synchronized manner. In case of humoral immune response, the exposure to antigen, it triggers to differentiate the plasma cell and memory cell from the original pluripotent immature bone marrow cells, B cells. And the plasma cell produce antibodies of all the classes. Antibodies are the uh, molecule, the protein rich molecule. They are looking like this. The stretches of amino acid is transcribed. The gene of immunoglobulin gene, or IgG, it is transcribed into the nascent mRNA and translated to the immunoglobulin protein. This antibody has a specific structure like Y shape. This Y shape has the variable region. These red portions are the variable region, means the amino acid sequence varies uh, very much within a species also. But there are rest portion called the constant region, this blue portion, and within a species, genus, species, you know the classification methodology of naming organism, maybe an animal or a plant. So there are genus, species, phylum, uh, subphylum. There are different classifications. So within a species, all the constant region amino acid sequences are almost same. That is why they are called the C or constant. Now there are V region variable, and there are two chains making up the whole antibody structure. One is heavy chain, these long stretches of amino acid. Another is light chain. The heavy chain and the light chain, they are interlinked with each other with a disulfide bond, and the two heavy chains, they are interlinked with each other with two disulfide bonds, and this region, it is called the hinge region. The hinge region gives the flexibility of the antibody structure, where the antibody can move 360-degree on these FAB portions, these Y hand portions. As we can move our hand, we can move our neck, head, to give the more exposure of the environment. For this reason only, the hinge region is made flexible, and the FAB portion can move here and there by fixing the constant region over a cell surface anchored. Now, how the immunoglobulin enacts as a ligand. The structure of the immunoglobulin is looking like this. The FAB is this portion and FC is this portion. This FC portion is having some complementary sequences of the receptor expressed over the host cell. Now, the host cell receptor amino acid sequence and the design of their orientation would match with the antibody FC region, so that they can fit with each other perfectly. Now, receptor and ligand mediated interaction between the pathogen and the host cell will be very firm, so that the blood flow will not just displace the antigen or antibody from each other. Now, the nature of the antibody, how the antibodies are made up, composed of. Antibodies are mainly the glycoprotein. It exists in monomers, dimers or pentamers. The basic antibody structure has 4 polypeptide chains, 2 identical light chain, 2 identical heavy chain. The region of heavy and light chains are variable region and the constant region. The FAB portions is displaying the N-terminal end, and the FC portion is C-terminal end. These C-terminal end can help them to anchor over a cell surface firmly, and N-terminal end help them to recognize different pathogen, epitope of the pathogen, and to get hold of that pathogen. So, the design of the paratube, these antibody site where the interaction of the epitope happens, it is called the paratube. You can see there are groups. This is the design of the paratube. The design of the paratube and the design of the epitope should match each other perfectly. As key fits with a lock, the matching of the key is needed to get the lock open. The same system is working here also. These are the antigen binding site, and this is the hinge region, and the constant region FC portion is the anchoring site. Now, the immunoglobulin, they are referred as the gamma globulin. Different names they carry: immunoglobulin, immunoserum immunoglobulin, gamma globulin. All are the synonyms of the immunoglobulins. The variety of the antibodies are, there are 5 varieties of the antibody: immunoglobulin M, immunoglobulin G, immunoglobulin A, and immunoglobulin D and E. Based on the structural differences in the constant region of the heavy chain, their characteristic differs and they are classified into these 5 varieties. If you see, they will be looking almost the same, but the amino acid sequence in their constant region varies, because in the constant region, if they varies, the immunoglobulin if varies, then the characteristic features will also change. They will be looking like this, their molecular organization. All are monomer, IgG, IgD, IgE, but IgA, immunoglobulin A, they are dimer. In their Fc region, the two monomeric immunoglobulin A are joined with a joining chain, J chain, and the IgM, immunoglobulin M, is a pentameric complex antibody, where the monomers of IgM, they are interlinked with each other and a joining chain facilitates this. So, the IgM is pentameric, IgG is dimeric, IgA is dimeric, IgG, D and E is monomeric antibody, which are prevailing in the nature. There are sub varieties of the immunoglobulin G, Ig1, 2, 3 and 4. You just see the two heavy chains, they are interlinked with the disulfide bonds. You see. Now, what are the function? This function is some critically depicted, but simply you just know the immunoglobulin M is the first line antibody secreted in the primary immune response. Means whenever the body interacts with the pathogen, the innate immune system starts. After the innate immune system getting triggered, the humoral immune response is the most important initiating factor. Now, what is happening? The initiation of the humoral immune response starts with the production of the antibody, and the first line antibody produced in the body is immunoglobulin M. As the immunoglobulin M is pentameric, naturally pentameric complex formation happens, there are implementations and implications also. At a time when the body faces the pathogen for the first time, when the antibodies are clumped together, they will be having more FABs to interact at a time. Form the basic idea. Here at a time, 5 immunoglobulin M molecules are interacting with each other, interlinked with each other. So, if pathogen interacts at a time, 10 FAB portions will be accessing the pathogen single molecule, and it would help to neutralize, to clear, to lyse, to kill the pathogen very fastly. That is why the first line antibody in the primary immune response is prevailing in the body system as a pentameric complex. Then what is the next level of antibody produced? The next level of antibody produced is immunoglobulin G. In the primary immune response, IgM is mostly prevailing. Next one is IgG. IgG also having some unique property. What is the property? You just see, crosses the placenta, plus means all the Ig varieties can cross the placenta, but other varieties can't cross the placenta. What is the placenta? Do you know? Anybody know what is the placenta?
Ma'am, connection between the mother and the fetus.
Yeah, that is called the umbilical cord, not the placenta.
I'm the hair-like
Placenta.
structures on cell, like
Ma'am,
cell
No,
from
no,
membrane.
no,
this
no, 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 no,
protoplasm.
no. No, 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 no. You are going to the right way only. But placenta is the covering, sac-like covering the fetus inside the mother's womb. And these placental membrane compose of a sac where the embryo grows to fetus, then a full-grown baby, and the fetus through the umbilical cord or the placental cord is joined with the mother. So, whenever the baby is in their gestational period, there is no other way to serve food, oxygen, protection through antibody, immunity, anything you can't give until and unless the placental cord is joining this fetus to the mother. And only variety of the antibody is immunoglobulin G, which can cross the placental barrier and reach to the fetus. You can just can have a idea how important it is? The whole year, almost 9-10 months, the baby is protected only through immunoglobulin G, which is produced in the mother and supplied to the baby. No other antibodies can reach, can cross the placental barrier. It is a very critical semi-permeable membrane derived sac so that pathogens can't reach there. Nature has created this one, and this variety of the antibody can only reach. Now, the other antibody population, that immunoglobulin E, immunoglobulin D and immunoglobulin A, these varieties of the antibody, they are mainly produced in the secondary immune response. Now, what is secondary immune response? Can anybody tell? What is secondary immune response?
Ma'am, I think it is white blood cells that production.
No,
No,
no, secondary immune response.
Ma'am, after the primary infection,
Yes,
again
yes, yes,
infection takes place. It is
yes. For the first time when body interacts with the pathogen, primary immune response is given. The cell is educated against the pathogen. What are the composition of the pathogen? What are the variety of the pathogenic molecule? Then one population of the cell is just trying to get effector functions and resolve the pathogen for that very time, and one population of the cells, maybe the B cell, T cell, all these cell populations carry some memory of that very pathogen, not all the pathogens. So this memory is stored through this cell as the memory cell. And for the second time, if the same antigen, not the different antigen, if the same antigen is interacting, body can readily recognize this one, readily can enact against the pathogen, secrete the antibody very fastly. No time is wasted, because in the primary immune response, 10 to 12 days time is necessary to react, to give the reaction. Like whenever you are reading a chapter for the first time, it needs some time na, to grab this one, and for the second or third time when you are just revising this one, you can easily grab the matter and reconcile the previous education. So this is called the memory. In the secondary immune response or in the tertiary immune response, the immunocompetent memory cell react and they give rise to these variety of the antibodies. That is immunoglobulin A. What do the immunoglobulin A do? What do the immunoglobulin do? They are the secretory type of antibody. That is why from sweat, from tear, from sebum, from milk, they are secreted, and through a secretory type of antibody, they can give protection to the body system or else. Now, what is the function? Main function, we are talking about the main function. Main function of immunoglobulin E, it takes part in allergic reaction. activates classical complement pathway, that is the innate immune response giving pro-protein complexes, and this immunoglobulin E, it helps to give the allergic reaction, induces mast cell degranulation and the release of histamines. That is why we take antihistamine for allergic actions. And the mode of action of the IgA is, IgE is the degranulation of the granulocyte. We have read what are the granulocyte. Can anybody tell what are the granulocyte population of the
Eosinophil,
cell?
basophil, and neutrophil.
Ah, good. Eosinophil, basophil and neutrophil. Let me go to that slide for others revision. Just see, these are the polymorphonuclear nucleosides, meaning their nucleus is polymorphogenic, having different shapes, multi-lobed, and they have dots. You just see they have dots inside their cytoplasm. What are these dots? You can't see these dots in the monocytes or lymphocytes or erythrocytes. What are lymphocytes? B cell and T cell. What are monocytes? Monocytes having the myeloid origin, and they give rise to macrophage, monocyte itself, mast cell, or the dendritic cell, as the erythrocytes are the red blood cells. So these granulocytes, they contain granules called zymogen granule. Whenever some infection, allergic infection happens, and the I won't tell allergic infection happens, some invasion of the pathogen happens, then these zymogen granule just releases histamines, and these histamines are the chemicals which in next step kills the pathogen, lyse the pathogen. It is a harsh chemical. So
But
the
ma'am, there is
immune
one doubt. Ma'am, if we take the yes,
Mhm.
ma'am. Ma'am, if we take the antihistamine, will it not interfere in the pathway of the
Yeah,
response? If
yes,
so, it
yes,
will.
very valid point to be raised, that that is why for triple reason we are having some sneezing, watery nose, watery eyes, tears are coming, or some rashes over the body. These are the signature of inflammation, isn't it? So whenever we are getting something allergic reaction, we should not take the antihistamine very easily. Please wait for 1 or 2 days, let the body fight, because it is the steps the body is taking to resolve the pathogen of their own. And if you just give the antihistamine, and they will render the histamine to be released from naturally occurring cells, granulocyte, then it would hinder the inflammatory pathway, your immune response. That is why only we should not take all the time in antihistamine in a very common cold or watery nose for this one, or allergic reaction. Now the IgD. What about IgD? You just search the plus. Where it is plus? Because in the function columns, everywhere it is negative. Where it is positive? Only present on the membrane of mature B cell. So the B cell, when they get mature, they express some antibody, they release the antibody, and the expression of the antibody over the B cell, here the antibody variety is mainly immunoglobulin D, and when this antibody expression production is in an optimum level, they are secreted in the blood or lymphatic circulations. So these are the antibody function and the variety we just considered. Now in the next level, disease and diagnosis, in immunological disease and diagnosis, before that one we will reconcile the slide number 16 and 17 and 18. Now what about this? The clonal selection of the lymphocytes. Now the lymphocytes, they are produced from a progenitor stem cell in the bone marrow, and clonally they have to multiply. Clonally means from a single cell to multiple cell. Genetically they will be same. From a single cell, multiple cells will be originated, divided and proliferated. Now if the cell is having their characteristic feature exactly same, then how these variety of the antigen will be recognized by the antibody, because the last step is desire to produce antibody only, because the main function of the B cell is to produce antibody. So antibody has their variety of 5. We read there are 5 antibody variety. Now you just think there are millions and trillions of antigen prevailing in the environment. These environmental antigen is numbers and the antibody is of only 5. Then how 5 will be just fighting against these trillions? What is the methodology? And what is the tricks nature has been already prefixed to make you very immune competent person? Then we would consider this one as a clonal selection theory. That is, B cell will be clonally expanded, but the B cell, if they don't interact with any antigen, they are uh, instructed by the brain that there is no need for your proliferation or increase in number. No need, because everything the body is doing, ATP consumption is needed. You have to put your energy, otherwise cell division, otherwise DNA replication won't happen. So why there is no infection? Why the body would waste its energy? No need. Just to survive, body would develop some bone marrow generated B cell, give the basic immune response, and it's enough. No need for their multiplication in numbers. So antigen interaction is the deciding factor. Now which antigen? Self antigen and non-self antigen. Why the self proteins are called as the self antigen? We all know antigen is the non-self product. We have read in the first slide. Self antigen is having its own meaning, where you would consider as a autoimmune disease. What is autoimmune disease? When the body fail to differentiate the self and non-self proteins products of the body, and the metabolites, and the self proteins are recognized as the non-self one, and the whole cell system immune response is triggered against this antigen, though it is the self antigen, and the autoimmunity is triggered. Now normally self metabolites, cell products, self antigens are not triggering the immune response. Only the foreign antigen. The design, can you see properly? Make it visible. The design, the amino acid sequence, the epitopes, because all the antigen will be interacting with the antibody in a epitope paratope manner. Don't forget, the epitope is the special site of the antigen which would only interact with the antibody expressed over the cell, the B cell. So B cell is expressing the receptor. What is the receptor? Can anybody tell? What is the B cell receptor? Now only we discussed. B cell receptor, immunoglobulin D. The immunoglobulin D is acting as the receptor, and the FAB portion of the immunoglobulin D a paratope will be interacting with the antigenic epitope. Now, when there is self antigen, the interaction is not given signal to get it triggered to clonally expand. But if there are foreign antigen, you just see the structure of the foreign antigen is matching with the receptor of the B cell. Pool of mature naive lymphocyte.
Ma'am, one doubt.
You, well,
Ma'am, this self antigen is there to like improve the innate immunity, to improve the existing immunity of the body. Because then why self antigens are needed?
no, self antigen are not needed. You won't say anything which are self as antigen. The definition of antigen is it is the non-self one. So self antigen means any self metabolite, any cell protein, carbohydrate, lipid, any portion which are recognized as their self product of the body. It is not meant to be triggering the immune system. We are telling them
Okay.
antigen, but they are not antigen. To be an antigen, the main criteria is to be non-self. Only in case where sometimes brain fails to recognize the self and non-self characteristics, and when sometime brain is failing, then the signal is got no, there is no discrimination, please divide, then take part in the immune response and all these things. That is why the self products which are not to be giving the signal to proliferate or increase in number, they are not a stimulus for antigenic response. But when the foreign antigen, which are a proper antigen, the body interacts, then brain gives signal, then yes, there is problem. Some foreign invaders are invading our problem will be recurred. So please get charged. Then only in the periphery, here all this function is going on. In bone marrow, B cell has been produced, they have been multiplied and developed and matured. In the thymus, the T cell maturing in the bone marrow coming to the thymus, they are matured here, and the cross talking between the B cell and T cell happens in the peripheral lymphoid organs. When this cross talking happens, the information exchange between the B cell and the T cell happens. What is the main function and main information the body gets? If any self product by mistake also it is represented to the T cytotoxic cell by the MNC molecule, then brain will check, no, by mistake you have taken it, or normally no such representation will be given. But in case of foreign antigen, the brain decides, that is, we call the brain is the sole factor which instruct all the signaling cascade, it should be on or off. All the metabolism going on inside our body, on the cell signaling cascade, all the cell cycle, they have their own feedback mechanism. In microbiology, you have read the respiration, TCA cycle, cyclic manner this has been producing. But all the cases inside the body, nature has created a feedback mechanism which can stop any reaction at any time when there is no need felt by the brain. Understood? So, in this factor, the clonal expansion happens from a single cell, multiple B cell is generating, it is called the clonal expansion. But the cell which is interacting with the foreign antigen, that will be proliferated. The cell which are interacting with the self products, they are not being proliferated, or proliferation means increase in number. So if these populations are not increased in number, then these antigen specific antibody will not be produced. and B cell only taking part in the humoral immune response. So this antigen cleaving methodology, mechanism of action is stopped. But when the foreign antigen is coming, the signal is given by the brain, okay, there is time enough to get it charged, and the B cell, which are only, there are different populations of the B cell arising in different lymph nodes. You just see, and which B cell is interacting with the foreign one, that one will be chosen naturally, normally in the peripheral lymph nodes, and they will be clonally expanded. From one B cell, the memory is being carried, which antigen they are interacting, not the A, B, D, C, no. Only if we name it as E, then the E antigen interacting with the B cell, that B cell population will be only proliferated, increase in number, differentiated to give specific activated immune response to form a clone of effector cells. Effector cells means for that time being the cells are being charged and they will be given rise to give immune response. Understood? So this is called the clonal expansion and clonal selection. At first clonal selection happens, which one is the desirable one, and then the clonal expansion.
Ma'am, can you explain the significance of helper T cells once?
Um, how many minutes we have? May I show a video? Then it will be very easy for you. I will be explaining, because in these PPT, because these PPTs are commonly made. So here I can't change. All the sections are reading the same PPT, but without the conception of T cell, you can't have a basic conception of immune response. So wait for a while, I will be playing one video. It would be very helpful how the total immune response is enacting. Is it okay?
Okay, ma'am.
Only T cell or the B cell T cell interaction do you need?
Ma'am, I think we can do it in next class because we have class from 12.
Oh,
Then.
okay. So I need one more class because for microbiology, अरे microbiology না না আরেকটা কি বাকি আছে বলতে আমাদের? What is left? Immunology, microbiology, cell biology. Microbiology I have completed. No, I think. So microbiology, for microbiology I need also one class. So can you give me one extra class whenever you are free? I don't have any problem, because for corona I lost 1 2 classes na. You please decide and let me know in weekend or any day you desire, I can take class. And please give me one class extra. T cell, I have more 3 minutes. So please have a look in a T cell interaction, B cell T cell interaction. A quick overview of the T cell activation. Just wait for 3 minutes only. Can you see the video? Just confirm me.
Yes, ma'am.
Yes, ma'am.
The antigen presenting cell, this one having some protrusion over this one, and this yellow one is the T cell. Now how interaction happens? The MHC expressed over the antigen presenting cell and T cell receptor over the T cell, they interact with each other. Other co-receptors, that B7 ligand and CD28 over the T cell, they interact. The step mediated activation of the T cell helps to multiply the T cell and interact like this. But the tumor can evade the immune system and T cell activation is seen. The receptor on T cell and APC regulate the T cell responsiveness. How they are interacting? If there is necessity, then only the proliferation of the T cell interaction will happen. This is the surface of the B cell or monocyte, and this yellow is the surface of the T cell. Now you just see there are different co-receptors. Main receptor is MHC and TCR. MHC is expressed over the antigen presenting cell, and TCR, T cell receptor, is expressed over the T cell. After this signal, the other co-receptor ligation happens. The CD40 is expressed over the antigen presenting cell and CD40 ligand will be expressed over the T cell. Likewise, different co-receptors are expressed and interacted with each other. This interaction of the receptor and co-receptor mediation helps you just see this PD1. PD1 is the immunoinhibitory receptor. So it acts as the feedback mechanism. Whenever there is no need for this triggering of cell signaling cascade on, then PD will be interacting and the immunoinhibitory functions will be expressed. Several immunoinhibitory functions are also there. CTLA4 also. They are ready to stop the signal. Whenever there is no necessity, the signal will be stopped. Activation of T cell is followed by application of natural brake. You see, the signal is passed. The interaction cross-talking happened. Here the antigen is displayed. This one is the antigen. The interaction happens, then signal is on. The activity and proliferation of the T cell is attenuated if there is inhibitory signal, and if there is the stimulatory signal happens, then the T cell proliferates. You can block the inhibitory signal also to get it charged. The aim is to improve T cell activity against tumor cell. How to improve? So can you have some idea? Next class we will be just touching the helper T cell as you pointed out, this one. or I can record this one for you and make it available there also. Okay. So today there is no time. Let me stop and take the attendance, please. Okay. Bye bye.
Thank you, ma'am.
Thank you.